after this we will be moving into transcription. Okay, so let's see. So in last class also I have shown you how this DNA damage can occur. Okay, so DNA damage can occur maybe due to single base alteration, or there may be two base alteration, or there may be chain breakage, or there may be cross linkages. For example, single base alteration means it may be due to deamination, conversion of purine into pyrimidine or pyrimidine into purine, or there may be two base alteration like due to UV dimer formation, or it may be due to chain breakage by free radicals, or it may be cross linkages be due to many other mutagens. So any DNA damage which leads to mutation. So we have discussed a lot about mutation. So there may be two type of mutation, namely point mutation and friendship mutation. Point mutation around a single nitrogen base. It may be transition or it may be transversion. Transition is purine by purine or pyrimidine by pyrimidine, whereas in transversion, purine is substituted by pyrimidine and the vice versa. Or there may be friendship mutation, so consequences may be silent mutation, or maybe missense mutation. So last time I am giving you the example in missense mutation, that is whenever GUG is converted into GAZ, at that time glutamate is replaced by valine. Glutamate is an acidic amino acid and valine is a neutral amino acid. That's why the biconcave shape of RBC is better held by the acidic unit, that is glutamate, which is replaced by valine in case of sickle cell anemia, hence the shape of the RBC is lost. So this is due to missense mutation. Sometimes there may be nonsense mutation where there is conversion of coding strand or coding code working code into a terminating codon like UAA, UAG and UGA. So there are different type of damage and its. So these are the ways in which it can be repaired. Okay, so here. So I think you are hearing me, okay? If you are not hearing, you can tell me. So I am talking about this thing, okay? So everybody is hearing my voice?
Yes, sir.
Okay, so let's continue, okay? So any type of damage or the four type of damage that we have discussed earlier can be repaired by the body itself, okay? There is a mechanism for the repairment of those type of damage. Repair mechanism is there, and that is called DNA repair mechanism. यस्तै भइरहेछ आज बत्ती जाने आउने इन्टरनेट बिग्रिने बन्ने. So it's going on in our NMC, okay? Leave that apart. ल अब मैले के भन्दै थिएँ भन्दाखेरि, so I'm talking about the repair mechanism. है सबैजनाले मलाई सुनिरहेछ होला. I hope everybody is listening. भनेपछि so four type of repair are there: base accession repair, nucleotide accession repair, mismatch repair and double strand breakage repair. र जुनसुकै रिपेयर गर्दाखेरि पनि एउटा बेसिक कुरा के सम्झिने भने फर्स्ट अफ अल द मिस्टेकन प्लेस इज आइडेन्टिफाइड. जहाँनिर मिस्टेक भएको त्यो ठाउँलाई पहिला आइडेन्टिफाई गरिन्छ. Then a section of the damaged DNA strand is cut down. एउटा भाग चाहिँ काटेर मिल्काइन्छ, त्यसमा चाहिँ नि now the correct one is synthesized and at last it is joined together. So तीनटा एक्सन आइडेन्टिफिकेसन गर्ने, त्यसपछि रिमुभल गर्ने र लास्टमा जोड्ने. यो चाहिँ नि इन एभ्री टाइप अफ रिपेयर मेकानिजम इट इज अ कमन फेनोमेनन. Okay, that is very interesting part. त्यति सम्झ्यो भने सम्झिन केही गाह्रो छैन. दिस इज फर एन इक्जाम्पल. ओके, यो सुरुको चाहिँ के हो त भन्दा इट इज अ नर्मल डिएनए एन्ड मिस्टेकनली के भएको थियो त भन्दा टिएजिसी हुनुपर्ने, यहाँ चाहिँ नि सरी इन प्लेस अफ जी देयर सुड बी ए, यहाँ ए हुनु पर्थ्यो होइन त, अथवा यहाँ चाहिँ नि यु सुड इज मिस्टेकन हियर, यु चाहिँ नि रिमुभ हुनु पर्थ्यो. So this U is first removed by glycosylase action. So by the action of enzyme, this racil is removed. त्यसपछि यो खाली भयो, इन्टु गरेको ब्ल्याङ्क प्लेस र अनि त्यसपछि now there comes the endonuclease. यो इन्जाइमको नाम सम्झिने, यसलाई रेस्ट्रिक्सन endonuclease RE अथवा simply it is called endonuclease. So the endonuclease is the genetic seizure. It acts as a seizure. It comes in the places where there is necessity of cutting the different strain or cutting the different section of gene. Okay, so that is endonuclease. So by the action of endonuclease there comes the empty place. यो सबै सेक्सन नै खाली भयो र यो खाली भइसकेपछि now the correct one is synthesized by DNA polymerase, namely it is DNA polymerase 3. र यो करेक्ट ए को अगेन्स्टमा टी, सी को जी को अगेन्स्टमा सी, जी को अगेन्स्टमा सी, ए को अगेन्स्टमा टी. now the correct one is synthesized and at last it is joined together by ligase, which is a genetic glue. भन्छ यो दुईटा इन्जाइम अथवा यो तीनटा इन्जाइम चाहिँ हरेक ठाउँमा हुन्छ, इन्डोन्युक्लियस, पोलिमरेज, लिगेज. यसरी के भयो त एनी रिपेयर द्याट इज done around a single base, a base excision repair is this mechanism. Okay, so I think it is okay for you. सबैले बुझ्यो होला यो. त्यसपछि if it is a nucleotide excision repair, अब यसलाई चाहिँ nucleotide excision repair बनाउनु पर्यो भने चाहिँ के गर्ने त भन्दा, so it may be due to UV light, ionization and other large scale damage. The first damage is identified, double helical is unwinded, damage is exposed, the excision nuclease cut damaged piece, polymerase and ligase fill the gap. यहाँ पनि के त्यही गर्ने हो जस्तो लेट्स सी यो चाहिँ नि डट 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 गरेको, दिस इज अ डिफेक्टिभ प्लेस यो चाहिँ एन्ड दिस इज आइडेन्टिफाइड. यहाँदेखि यहाँसम्म चाहिँ डिफेक्टिभ पिस हो, यदि काट्नुपर्छ भन्ने कुरा आइडेन्टिफाइड भयो. देन इट इज कट बाई इन्डोन्युक्लियस. यो चाहिँ नि के भयो त नाउ दिस डिफेक्टिभ पिस इज कट डाउन. यदि कट डाउन भइसकेपछि के देख्न सकिन्छ त भन्दा फ्रम हियर टु हियर यो दुईटा ब्लू लेटरको एकै कलरको बिचमा खाली ठाउँ भयो. सो दिस एम्प्टी प्लेस दिस इज रिमुभ. यो चाहिँ यति काटेर पठायो, यो चाहिँ यति खाली ठाउँ भयो. दिस खाली ठाउँ इज फिल अप बाई पोलिमरेज, देन जोइन बाई लिगेज एन्ड इट इज रिपिट. यसमा पनि के भयो त फेरि सम्झिने तीनटा कुरा. फर्स्ट आइडेन्टिफिकेसन अफ द डिफेक्टिभ प्लेस, देन कटिङ, देन पोलिमराइजिङ एन्ड जोइनिङ. सो दिस इज अबाउट न्युक्लियोटाइड एक्सिसन रिपेयर. यसमा चाहिँ some diseases are associated with inadequate or inefficient nucleotide excision repair mechanism such as Ganoderma pigmentosum, ataxia, telangiectasia, Fanconi's anemia, Bloom syndrome, Cockini's syndrome, SPCC, that is hereditary polyposis colon cancer. So many types of diseases are there which are associated with ineffective nucleotide excision repair. Okay, so this is about nucleotide excision repair. Then mismatch repair. So here also, so what is mismatch repair? So for repair mismatch, for example, C to A or T to G, template strand is first methylated. So methylated is recognized. Enzyme GATC endonuclease, or mainly let's say endonuclease cut the strand at the adjacent methylated at the adjacent methylated GATC sequence, defective strand is digested by endonuclease and new one is correctly synthesized by polymerase and ligase. यसमा पनि त्यही गर्ने हेर यसमा चाहिँ एउटा बडीले के गर्छ भन्दाखेरि this is generally seen in a higher organism. So how to identify the template strand and the defective strand? यसको लागि चाहिँ नि the body itself has a mechanism to methylate the correct strand or the template strand. You can see that in 3,5-5' this above in the above strand, this is methylated at two places. You can see two place methylic group is there. So it means this is the template strand. It need not to be cut. So the adjacent strand corresponding to the methylated strand is cut. So this is cut piece by exonucleases. So and this is the empty part, and this is correctly filled by polymerase and it is joined by ligases. So here also mechanism is same: identification, removal, then the polymerization, then the ligation or the joining. In this way mismatch repair can be repaired or mismatch damage can be repaired. Okay, so defective mismatch repair or HNPCC. We have CSPCC or hereditary polyposis colon cancer is related with the nucleotide excision repair, and this thing a mismatch repair is related with the hereditary non-polyposis colon cancer. So this is this. And about the fourth one, okay, so whenever there is double strand break repair, so much detail about double strand break repair is not given in books. It is a recent phenomenon. Still, we can say that it's a dangerous condition. It may result in chromosomal defect and cell death, and it is repaired by homologous recombination like in yeast, or non-homologous end joining like in mammals. So it is done by joining of the similar or like you have seen in the let's say in the cell division, in the cell division ma चाहिँ एउटा सेल माथि अर्को सेल बसेर homologous non homologous pairing बनाएको जस्तै यसमा पनि there is homologous recombination, and by this mechanism the double strand break repair is done. So this is about 4 different type of DNA damage and repair: base excision repair, nucleotide excision repair, mismatch repair, and double strand break repair. So in the examination, please draw these figures. Okay, यो सबैले screenshot गरेर राख्ने. If you are writing, you can write it down also. र यो सबैले यसको बारेमा लेख्ने. This is generally asked in 3 marks question. यो tail question को रूपमा धेरै सोधिरहने question. Okay, whenever we are asking question from replication chapter, generally the question will be 2 part. One is about the replication fork and another is about the DNA damage and repair or mutation like that. So these are the probable questions. And if you see the differences between prokaryotic and eukaryotic process, mainly this replication process, you can find many differences. So some important ones are listed here. So if we compare the RNA primer length, it is around 50 nucleotides long in prokaryotes, which is 9 nucleotides long in eukaryotes. So it is shorter in case of eukaryotes. A type of polymerase is DNA polymerase 1, 2, 3 in prokaryotes, whereas in eukaryotes it is DNA polymerase alpha, beta, gamma, delta, and epsilon. So 5 types are there. Number of origin is single in prokaryotes and it is multiple in eukaryotes. Length of Okazaki fragments may be many, maybe from 1,000 to 2,000 in nucleotides, whereas it is very less in comparison to prokaryotes, around 200 in eukaryotes. So in eukaryotes, around 200, in prokaryotes is around 1,000 to 2,000 nucleotides. And if you see the rate, it is very, very fast in prokaryotes. Around per second, there is 500 nucleotide adhesion, which is much slower, 10 times slower in case of eukaryotes due to mechanical complexity. 50 nucleotide per second is added. So this is the rate. So these are some differences, basic differences between prokaryotic and eukaryotic replication. So this is all about replication. So I hope everybody is now aware with the different type of replication. Okay, so these are the same thing. Okay, so you can also see here there are base accession repair, nucleotide accession repair, homologous repair, mismatch repair. So how it can cause, you can see maybe due to hydrolysis or oxidation or alkylation or UV radiation, or due to hydrolysis, oxidation, alkylation. This is accession repair, which is very common. Okay, 10 lakhs per day. So 60 per day is nucleotide accession repair, 60 per day is ionization repair, and around 600 per cell division after proofreading mismatch repair. So it is done frequently in the body. Okay. So you can see that polymerase makes a mistake about every 1 in 10,000 bases. That is, so are you listening? मेरो कुरा सुनिरहेछौ?
Yes, sir.
Okay. Okay. So ionization radiation, it may be or replication error. So you can see the causes here. You can see how they are repaired here, and you can see some additional feature about this thing. Okay. So more time cell device more likely to have mutation in something important. So mutation phenomenon, it is everyday phenomenon. Most of the time it is corrected, sometimes it gets inherited and becomes mutation. This is also this thing how they are repair. We have already discussed about all the thing. Okay, so it may be mismatch repair, or direct reversal, or repair of single strand break repair, a nucleotide accession repair, or non-homologous in-joining. So different type of repair mechanism are given here. Okay, homologous joining, non-homologous joining. So it is about 4 type of repair mechanism where the similar strands are put together and they are joined after removing the defective piece. So this is about molecular mechanism. So this is also overview. Okay, how DNA damage can be done. It may be done due to endogenous agents such as diet-related bile acid or macrophage or neutrophil, or production of reactive oxygen species or nitrogen species. So it may be due to free radical. Okay, so it may be exogenous agent, maybe due to smoke, radiation, metals, viruses, and other genotoxins. So they may all lead to DNA damage. So DNA damage may leads to change in it may lead to somatic mutation, or it may lead to epigenetic alteration, or it may lead to germline mutation. So anyway, they all leads to different type of mutation, which ultimately progress or multiply, alter, field detect with driver mutation, and finally it may be, if it is not treated, if it is not repaired, or if DNA damage is deficient, then there is increase on repair DNA damage, lead to increased somatic mutation and epigenetic alteration, and finally leads to cancer. So it is one of the major cancer region of cancer is inability of the body of the cell to repair the DNA damage. All this mechanism we have discussed earlier, if this is going to occur frequently or with the constant rate, then there is less chance of cancer. If not, there is high chance of cancer. So these are some diseases which are related with the defective DNA repair mechanism. Okay, we have discussed, we have already seen the example. So let's see some of the examples. So HNPCC, then colorectal cancer, then genoderma pigmentosum, then Bloom syndrome, Fanconi syndrome, hereditary breast cancer, BRCA1, BRCA2 deficiency. So these all are different type of disease, which may be due to it is given DNA mismatch repair, nucleotide excision repair, or it is defect in the double strand break. So all these things are there. Okay, so cancer susceptibility symptoms, everything is provided here. So this is very informative chart, you can see, and you can go into the detail of all these things. But from the examination point of view, please remember this 4 heading. Okay, you have to, you'll be asked to write about write short note on DNA damage and repair, or what are the repair mechanism at that time. You have to mention this thing, and you may not discuss about mutation because it is asked differently. So you can start from here. So there may be 4 type of damages: single base alteration, 2 base alteration, chain breakage, chain leakage. Then you can write about the repair mechanism, and every repair mechanism, please draw the figure. If you remember, write about some diseases like that. Okay, so this is about replication and DNA damage and repair. And then we can slowly go into our next chapter, that is transcription. Okay, so everybody is hearing me? Sabaile sunira chhau? Yes?
Yes, sir.
Okay, very good. So let's start the transcription. Okay, so transcription is much easier than the replication. So first of all, transcription is a genetic process, and it is a continuation of central dogma of life, or it is also some book. It is given as the first phase or first step in translation. So it is also a part of protein formation. कुनै कुनै बुकले के गरे हुन्छ अरे भन्दा ट्रान्सक्रिप्सनलाई छुट्टै हेडिङमा नराखेर it is given as a first part in translation process, but traditionally it has been kept as a separate heading. Okay. So transcription is a process in which RNA is produced from DNA, and this is not only. Yes,
सर स्लाइड चेन्ज भएको छैन.
स्लाइड चेन्ज भएको छैन. ट्रान्सक्रिप्सन भनेर लेखेको छैन.
छैन, डिएनए रिपेयर डिफेक्ट्स देखाइरहेको
तिमी
छ.
स्लाइड चाहिँ देखिरहेछौ.
हजुर.
You are not seeing the slide.
स्लाइड देखिरहेछ तर पहिलाकै च्याप्टर डिएनए रिपेयर डिफेक्ट्स कजिङ डिजिज.
हो र, ल पखै जस्ट अ सेकेन्ड. Now you can see it.
Yes, sir.
Okay, so let's continue. So transcription is a process of formation of RNA from the DNA, and it is not simply the production of RNA only, because RNA production itself is the first step in protein formation. So RNA is never produced by DNA unless and until there is necessity of protein formation in the body. कुनै जिनले चाहिँ नि कुनै प्रोटिन बनाउनु पर्यो भने फर्स्टमा के बनाउनु पर्छ त भन्दा फर्स्ट अफ अल आरएनए फर दैट प्रोटिन मस्ट बी फर्म. So whenever there is necessity of production of any protein in the body, at that time transcription occurs. So transcription is generally a production of RNA from DNA during the protein formation. So the synthesis of mRNA from the DNA template, which occurs in 5' to 3' direction. So every synthesis in genetics occurs in 5' to 3' direction. During transcription, a DNA sequence is read by an RNA polymerase, which produces a complementary and antiparallel strand, RNA strand. And transcription is the first step leading to gene expression. Gene expression leading to gene expression means that is production of protein. Okay. So this is it, okay. So see the figure carefully, okay. You can see this is the DNA. Any of these two strands of DNA, any strand can act as a template strand for RNA synthesis. But RNA synthesis occur only in a single strand, unlike the replication. In replication, in both the strand, a new piece of DNA is synthesized. But in case of transcription, only one strand is acting as a template strand. So this is a template strand, this is a coding strand. This is the opposite strand of template strand is called coding strand. This is called coding strand because the sequence in this newly formed RNA and this will be same. This will be the code for this RNA. Only difference is that in RNA there is uracil, in DNA strand there will be thymine. यो भन्दा बाहेक के हुन्छ त यो स्ट्रान्डमा जे छ एक्ज्याक्टली सिमिलर चीज छ नि यु क्यान सी इन द टेम्प्लेट स्ट्रान्ड खाली के फरक छ यसमा टी हुँदैन टी को ठाउँमा ए हुन्छ that's why it is called template strand. This is called coding strand. So this is whole is called transcription bubble. यसलाई के भनिन्छ ट्रान्सक्रिप्सन बबल भनेर भनिन्छ. So before that, okay, त्यो भन्दा पहिला चाहिँ नि let's talk about different things, different materials that are necessary for the RNA synthesis. पहिला त हाम्रो requirement हेर्नु पर्यो के चाहिन्छ भनेर. Okay, so first of all DNA template is required because without template we can't imagine about the RNA production. So transcription is mainly started whenever there is availability of template. So a single strand of DNA acts as a template to direct the formation of a complementary RNA transcript. Then a strand that is transcript to RNA molecule is referred to as template strand of the DNA. The other strand is referred to as coding strand of the gene. And substrate. So substrate, the raw material for RNA production. RNA is a ribonucleic acid. So it is a polymerized form of ribonucleic nucleotide. So R, ATP, GTP, CTP, or let's say AMP, GMP, CMP, UMP, they are the substrate for RNA production. And beside that, enzymes are required like DNA polymerase in replication. There is RNA polymerase in case of transcription. In transcription, it is done by the polymerization is done by RNA polymerase. So it is also called RNAP, and it is responsible for the synthesis of RNA in 5' to 3' direction using DNA as a template. And beside this RNA polymerase, different factors are also there in initiation factor, elongation factor, terminating factor. So different other type of special type of proteins are also used or utilized during the transcription process. And this is how RNA polymerase look like. Okay, and it is generally asked in your Viva examination, what is holoenzyme and coenzyme form of RNA polymerase? Or please remember that this RNA polymerase is a tetrameric protein. It has 4 polypeptide chain, 2 alpha, 1 beta, and 1 beta dash. 4 polypeptide chain is there. So any RNA polymerase having only 4 units is a core enzyme. It is not a holoenzyme. Holo means whole, complete. So to make the core enzyme, holoenzyme, there is addition of sigma factor. You can see this sleeping 6 here in between beta dash and 2 alpha. You can see the sleeping 6. This is called sigma factor. Okay, so this whenever sigma factor is added to the core enzyme, it becomes holoenzyme. So sigma factor holoenzyme, no sigma factor core enzyme. So this is very important. This has been the favorite question for your fiber examination. This is frequently asked question. Please note it down. Okay. So RNA polymerase do exist in two form: holoenzyme form or core enzyme form. In holoenzyme form, there is only in core enzyme form there is only 2 alpha beta dash, only 4 tetrameric sections are there. Whereas in case of holoenzyme, one additional factor is also there, which is called sigma factor. So this is about RNA polymerase. So like replication, this is also divided into 3 steps, 3 phases. Okay, one is initiation, and another one is elongation, then another one is termination. Initiation, elongation, and termination. So initiation is the starting, elongation is the making the template, and termination is dissociation of the ones form new RNA from the scene. So initiation in the nucleus, RNA polymerase recognize the recognition site. It is bind to the promoter site. The RNA polymerase then separate the DNA into single strand. So the template strand can be read in 3' to 5' phase. So what does it mean is that for initiation, there must be identification of the initiating site. So like in replication, we have term it as the origin of replication, ORI. In transcription, the initiation is done at the place where it is called promoter region. So there are different type of promoter site in case of prokaryotes, different type of promoter site in prokaryotes and eukaryotes. So it is called promoter site. So the first thing that occurs in promoter site is addition of holoenzyme form of RNA polymerase. So the main function of sigma factor is to carry that RNA polymerase to the promoter site and bind it, and that sigma factor will be again free. So again I am repeating, the sigma factor will take that RNA polymerase to promoter site, make it bind to the promoter site, then it is set free. Then by the action of RNA polymerase, there is complementary base pairing on the template strand of the DNA, and a new piece of RNA is formed, that is elongation. And ultimately, whenever there are 2 types of terminating processes are there. One is called row dependent and one is row independent. So RH row is a special type of protein like sigma factor. It is a terminating factor. So sometimes it is done by the addition of row factor, sometimes it is done by without addition of row factor. Okay, so before that, in shortcut, so there are 3 type of 3 steps. One is initiation, one is elongation, one is termination. So in initiation there is promoter site. In promoter site, sigma factor will take this RNA polymerase, bind it to the there, then this sigma factor is released. Now there is initiation, now there is elongation, there is opening of this strand, there is action of RNA polymerase, there is complementary base pairing, then there is formation of new RNA. This is elongation, and once elongation is done, at some time it may be due to the addition of row factor, or maybe without addition of row factor, there is termination process. So in this way, transcription is done. Okay, so today we have seen about initial steps of transcription. Okay, so today we'll keep up to this. Okay, and in next class I'll be completing your transcription, and after that transcription we will see post transcriptional modification, because the nascent RNA formed here is not the functional one, then it requires some sort of modification to convert that into the functional one, that is mRNA, rRNA, tRNA, and different type of other modifications are there. So for now, please remember about this thing: transcription, definition, how it is done, what is holoenzyme, what is core enzyme, then the steps, initiation, elongation, termination. Okay, and detail of all this thing we'll discuss in the next class. Okay, so for today I'm going to keep up to this. So if you have anything, you can ask me. So do you have any question?
Excuse me, sir.
Yes.
Double strand break repair को फेरि एक भनिदिनु न.
The double strand break repair. Just wait, okay. Okay, double strand को चाहिँ नि यसै गरी हुन्छ भनेर खासै त्यस्तो डिटेल मेकानिजम दिएको छैन. ओके, बट हामीलाई चाहिँ के भनेको छ भने इट मे बी डन बाई जोइनिङ होमोलोगस अर नन होमोलोगस एन्डिङ भनेर भनेको छ. सो इट इज अ टाइप अफ माइटोसिस अथवा मियोसिसमा चाहिँ हामीले क्रोमोजोम चाहिँ नि सिमिलर क्रोमोजोमहरू एकमाथि अर्को बसेर त्यहाँ चाहिँ नि एक्सचेन्ज अफ पिसिएस भएर चाहिँ नि जुन हाम्रो भेरिएसन आउने टेन्डेन्सी हेरेको थियौँ, त्यस्तै प्रकारको रिपेयर मेकानिजम चाहिँ यसमा हुन्छ भनेर चाहिँ वी क्यान गेस इट, बिकज इट इज कल्ड होमोलोगस एन्ड जोइनिङ प्रोसेस अर नन होमोलोगस एन्ड जोइनिङ प्रोसेस भनेर दिएको छ. भने हामीले चाहिँ नि त्यतै धेरै डिटेलमा त छैन, तर यसरी बुझ्दाखेरि चाहिँ होमोलोगस रिकम्बिनेसन गरेर अथवा होमोलोगस एन्ड जोइनिङ गरेर चाहिँ नि द डबल स्ट्यान्ड ब्रेक रिपेयर क्यान बी ड्यामेज क्यान बी रिपेयर भनेर भन्ने. ओके, यसको बारेमा अझ डिटेलमा पायो भने अझ डिटेलमा पनि हेरौँला. ओके करुणा?
ओके सर. सर अनि जर्म लाइन म्युटेसन भनेको के हो?
जर्म लाइन म्युटेसन भनेको चाहिँ नि अब दिस इज अल्सो अ टाइप अफ म्युटेसन. ओके, अब जर्म लाइनमा भएको चाहिँ नि एउटा जेनेरेसन गरेर चाहिँ नि पासन हुँदै हुँदै जान सक्छ. इट इज लिटिल डिफरेन्ट फ्रम अदर टाइप अफ म्युटेसन. ओके, जर्म लाइन सेल भन्ने नै छुट्टै प्रकारको सेलहरू हुन्छ. ठिक छ, वी क्यान डिस्कस later on. Okay, म तिमीलाई अझ यसको बारेमा अर्को क्लासमा फेरि भन्नु. ओके
ओके सर.
Okay everybody, so for today we'll keep up to this. Next class we'll continue. Thank you everybody, have a great day.
Thank you sir.